Hello, this is Mr. McMurray with your next exciting news of an assignment that you need to do for anatomy. Alright, this is the Cell Parts is Parts worksheet. Now, you have a picture here, and another picture, and another picture, and another picture, and so on. Until you have 6 total pictures. Now, we said that every cell is a little bit different. What I'd like for you to do on this assignment is to see some of the different cells and different drawings of them, and try to find the main features again that we've learned already. Okay, so first of all, I would recommend finding things like, oh, there's the nucleus, okay, and then going from there and finding the part you can. And so it's all about narrowing it down to the ones that you can't figure out. Okay, now a couple things. One, I would like you to actually type the name in. Don't whine. Some people had to whine about having to write names in, so I don't know if it's better or worse for you to type them, but you do need to type the names out over here by the labels. Now, some of them may be a little crowded here. You'll have to draw a little line maybe up here, and then a text box that labels E, and then another one here, or something like that. But please do not just write the letters over on this side. Okay, type the name out. Okay. Some are possibly a little difficult. For example, D here is pointing to that little hole right there. E is pointing it's this whole section here because it goes past and the line goes a little past it there. So that would point to this whole thing here. And I will put a little slack on that kind of thing. Now, if you'll notice, some things have 2 words like lysosome or peroxisome. In other words, it could be either one, so just label it either one and that will be fine because it's hard to tell them apart. Now, talking about lysosome and peroxisome, notice you'll also have vacuoles. Vacuoles and lysosome peroxisome look similar. They're just circles with a little bunch of little particles inside that are either being stored in the case of vacuoles, or they're being digested in the case of lysosomes. They're broken down. So, generally speaking, vacuoles are going to be larger. So V, for example, here is the biggest thing floating around with stuff in there. So that would be, for example, a vacuole. Smaller one is going to be a lysosome. Now, vesicles, for the most part anyhow, are going to be either coming or going, or they're fusing with the membrane and spitting something out, or the membrane pinching in and making a vesicle. So we're going to go with those vesicles right there. Alright. Sorry, it's about 4:50 in the morning and I'm starting to stay awake here. So, anyhow. Now, all the words over here will be used at least once. Some of them may be used more than once. So it's possible for them to use more than once. All of them will be used at least once. So like I said, label the ones that are the most obvious, and then you have to look through the back. Some hints also, rough ERs are always going to have little bumps or ribosomes on the outside of them. Okay. Smooth ER is going to look similar, but not the bumps. Smooth ER sometimes tends to be drawn more like a bunch of pipes. So in some cells, rather than a maze-looking thing, which is what it means, it looks more like a bunch of pipes, or like a water park nowadays with little pipes that you go through. So it looks more like that sometimes. Remember the Golgi apparatus, which is also smooth-looking, it looks more like a stack of pancakes that have been sliced in half. Okay. Now, some of these, for example, mitochondria and Golgi apparatus, we usually see in a picture where they've been sliced open. But you got to keep in mind, you might see them from different angles or just see the outside. So, the mitochondria might be a long, ovally shaped thing, and it will not always be cut open and have a little delay here. Okay, or you could see it from a different angle. You might see a stack of pancakes from the top. Okay, like a stack of that kind of frilly pancakes, you know, around the edges, little things that are pinching off and stuff. So it's not just solid pancakes. They might look like a stack of pancakes that is coming off a little bit. But anyhow, so if you'll go to that and do that for each of the pictures, okay. Good news is, since there are many, many pictures, that each one's worth like 1 point apiece or something. So you can miss quite a few and still have a pretty good grade. So if you'll go through, do the very best that you can. And once again, as always, if you have any questions, you can email that to me or call or whatever. All right. Enjoy. Alright, one last thing I forgot. Just for the record, on the tests, quizzes, etc., you will only have questions about the drawing that you have already learned from, okay, that we took the notes on. None of these cells will appear on the test. Okay, this is just kind of a way for you to try to learn to recognize these different things in different cells. They look a little different and they're drawn a little different. All right, but yes, on the test we will only use the original drawing that you had with the notes on it. All right, that's everything I